Hello everyone, my name is Nikhil Sahajpal. I am a postdoctoral fellow at Augusta University. I would like to thank the CGC Review Board for giving this opportunity to present our study at this forum. I will be talking about next generation cytogenetics or optical genome mapping, a proposal for cost-effective approach for comprehensive testing of prenatal cases. The current landscape of prenatal testing as recommended by ACMG, ACOG, and IACOG is that non-invasive prenatal testing is the standard of care, and for positive NIPT screens, high-risk pregnancies, and abnormal ultrasounds are recommended for invasive diagnostic testing using either amniocytes or chorionic villi samples. The recommended invasive tests include karyotyping, FISH, and chromosomal microarrays, which are either performed simultaneously or in tandem, as each technology has its own advantages and respective limitations. Karyotyping is ideal for whole genome structural variation detection at a lower resolution. However, the SV location and sizes remain inaccurate and some translocations remain cryptic. FISH panels are targeted, still the microdeletions and duplications remain difficult to detect. Chromosomal microarrays are ideal for copy number variation detection. However, they are limited in their ability to detect balanced translocations, inversions, and low-level mosaicism. With this in purview, I would like to dive into the current scenario of prenatal care. A screen-positive NIPT or high-risk pregnancy recommended for invasive prenatal testing. CVS or amniocentesis is performed, the cells are cultured, and a stat FISH or karyotype shows a normal profile. However, a segmental deletion is observed on chromosome 7. Now, if the CMA was performed simultaneously to karyotyping, it adds to the cost of the test, and if it was performed as a follow-up after a normal karyotype profile, it adds to the time to reach a diagnosis, which is actually very critical in prenatal care. So this raises a question, could we reach a definitive diagnosis in one step with one technology, assay, and analysis? This is the same case with the data using optical genome mapping. On the circus plot itself, we can see a deletion on chromosome 7 and a duplication on chromosome 2. The events can be seen at the copy number track and also as the SV level track. Similarly, the chromosome 2 duplication can be viewed at the CNV track and the SV track. So to answer my question, yes, we can reach a definitive diagnosis in one step with one technology, assay, or analysis. But probing further into this case, the duplication on chromosome 2 was not called out by microarray, whereas it was uniquely called out by optical genome mapping, as seen in this zoomed in view of the CNV track. Further, in the SV view, we can actually delineate the orientation where this duplication is in tandem orientation. Moving to the Sapphire workflow, it relies on the isolation of high molecular weight DNA from either amniocytes or chorionic villi cells. Once the DNA is isolated, it is labeled at particular 6 base pair motifs, which are loaded onto the Sapphire chip and goes into the Sapphire instrument. Inside the instrument, the DNA linearizes on the basis of electrophoresis and images are mapped. These images are converted into molecules which are used to form a consensus genome and is oriented against a reference genome to cause structural variation. A unique coverage based algorithm is used to call copy number variations. In this study, we have mapped so far 28 prenatal cases with one as trio. All cultured amnio samples had orthogonal testing, which included karyotyping with FISH or CML. The samples tested with OGM were blinded to the cytogenetics data, and accuracy and precision was measured at the level of sites, operators, and instruments. I will be discussing few select traces, which includes common trisomies such as trisomy 21, trisomy 13, sex chromosome aneuploidy, and will move to complex structural variation cases, following up with other SV classes such as trisomy 8. Case 1 is an example of trisomy 21, which is the most common live-born trisomy. Here in the circus plot itself, we can see trisomy 21 and a copy number gain on chromosome 15. The same event can be viewed at the CNV track. For those who are used to looking at microarray data would appreciate this CNV track, where we can see the copy number gain on chromosome 21 and on chromosome 15. Again, you can zoom into these chromosomes to see a clear view of the copy number change. The case 2 includes a case of trisomy 13, where we can see on the circus plot and also a copy number loss on chromosome 15. Similarly, the trisomy 13 can be viewed in the CNV track, and the deletion on chromosome 15 can be viewed in CNV track, and also in the SV maps, where we can see actually this is a homozygous deletion. Case 3 is an example of Mosaic Turner syndrome, where we can see in the circus plot, but a better representation of the mosaic CNV loss of chromosome X can be viewed in the CNV track representing Mosaic Turner syndrome. Moving on to complex structural variation case. Here we can see on the circus plot that there is a duplication on chromosome 15 with rearrangement and a translocation 1521. The zoomed in view shows the copy number gain on 15, the rearrangement, and the translocation 1521. The copy number gains can also be viewed at the CNV track. If we look more closely at this particular chromosome, we see that there are two segments with a CN state of 3. In the SV view, what we see is actually that these two segments are fused. And if you zoom into this particular map, we can see that the labels on this map are actually inverted. So what it represents is, if you consider this arrow as the chromosome segment with a CN state of 3, and this segment with another CN state of 3 is actually inverted and fused and continues in this orientation. For the sake of simplicity, you can derive your own ideograms, and here we have represented chromosome 15 as segment A, B, C, and D, where segment A and C are with the CN state of 3. Now translocation 1521 is actually easily to delineate, as this segment D translocates to chromosome 21 and forms derivative 21, whereas the CN state 3 and inverted foldback fusion into this chromosome results in derivative 15. When the data was compared with karyotype and fish, it was found to be 100% concordant. On the karyotype, the two arrows shows derivative 15. And moving on to the next case, which is an example of trisomy 8, and we can see an additional gain on chromosome 13. Again, the CNV track shows us the trisomy 8, but what we can see here is on chromosome 13 that there is a CNV track calling a gain, and the SV track shows us that this segment of the chromosome is duplicated. However, this region is actually uh, incorporated in an inverted orientation between this tandem duplication. Thus, it is able to resolve the structure much better. Again, the karyotype showing trisomy 8, which was concordant. Now, 5 samples were run at 2 different sites, which led to 100% inter-site, inter-operator, inter-instrument, and data concordance. So to conclude, invasive prenatal testing is recommended after a positive NIPT screen for high-risk pregnancies. Karyotype FISH and CMA are the recommended tests employed globally. Optical genome mapping has the potential to identify unique genomic abnormalities and also better characterize the SPs. OGM data was found 100% concordant with as compared to karyotype with FISH or chromosomal microarray. The data is actually our part to lab developed test, and it has certain advantages such as reduced hand-on time, faster turnaround time, and is cost effective as compared to the combined cost of karyotyping FISH. And with this, I would like to acknowledge the Director of Molecular and Cytogenetics Lab, Dr. Ravindra Kole, Dr. Ashish Mondal, and Colin Williams, our collaborators at University of Mississippi, Dr. Ferribos, Rashid Colvier, Dr. Suzanne Hurley, collaborators at Deadwood Genetics Center, Dr. Bob DuPont and Dr. Timothy Fee, and our optical genome mapping support team. Thank you so much for your attention.